Swayam Prabha. Digital India. Educated India.
Hello students, welcome to the course of organ printing. So till now we have discussed what is bioprinting, what are the different types of bioprinting techniques available. So in this lecture we'll discuss inkjet printing technology or inkjet 3D bioprinting technology. So inkjet printing is a very common technology that most of our offices we have printer that where some of the printers work on this inkjet principle. So in this case, in this printing, what happens here? Basically, the liquid droplets are produced, those of our controllable size and small volumes, and then those droplets are delivered or positioned or printed to a specific coordinates of a substrate or on a printing stage. So that this printing technology here, the main principle of this technology is the production of tiny controllable size of liquid droplets, right? And this technology is a low cost technology because most of you have seen that the printers are not very expensive. So most of our inkjet 3D printing, 3D bioprinting actually done by modifying the office printers to suit printing biomaterials, cells, and pharmaceutical components, right? So here, this printing technology, it is the cost is not very high. So it is a low cost technology, but it is highly efficient because some of the printers are capable of printing or producing liquid droplets of several hundreds per second. So this is a very efficient technology, very high throughput technology, and it's highly precise because these droplets of controllable size of repeatable or reproducible size of droplets can be easily produced with this technology, and various types of biomaterials or pharmaceutical formulations can be used for printing or for generating droplets so that we can use them for our printing purposes. And this inkjet printing is capable of producing droplets of the order of 1 picoliter. So it's a very fine or very tiny droplets can be produced. So the resolution of this printing technology is very high. So because we can produce this very tiny droplets and then we can plot them, or we can basically deliver them at certain coordinates of a printing structure, of a printing substrate or printing stage. So that's why this printing technology is highly popular because of all these phenomena, like all these advantages or all these characteristics. Like it is a low cost, high efficiency, high precision, and high resolution. Another very important characteristics of this technology is the drop on demand. So this technology is like whenever there is a requirement of a particular drop, requirement of a drop of a particular material, that time only the drop will be produced. So we can control the droplet generation and further their delivery those droplets to the particular coordinates for printing purposes. Another important characteristics of this printing technology is non-contact delivery, material delivery. Because here what happens, the printing nozzles and the stage are not connected, and they are not connected to the materials also, right? The drops are produced, those discrete separate droplets are produced from the nozzles, and those droplets are being delivered or printed on the printing stage. So nowhere these two, the printing stage and the nozzles, are coming in contact with each other. So this is a non-contact delivery. It has huge advantages for printing cells, printing pharmaceutical formulations, and also printing different other things where because due to contact there can be contamination. So that can be avoided by using because of this non-contact phenomena. So this is a very much a very good property for cell printing, because we can reduce the contamination using this kind of inkjet printing technology. So here, now with the initial success of inkjet printing led to use of this technology for bioprinting or 3D bioprinting, where what happens, where we are, our intention is to print biological relevant structures and function, biological relevant structures, so that we can generate or achieve certain biological functions by manipulating, patterning or assembling of biological relevant materials with cells or without cells, and also with biomolecules in a predefined and pre-designed manner to make up the 3D structures, so that we can get a biological relevant structure so that we can generate biological functions. So for all this, inkjet 3D bioprinting is very much used to develop this kind of 3 dimensions or tissue or organ structure using this technology. Here, the capability of this printing technology, it can deliver different types of materials and cells. So we can use, basically we can use different types of materials and cells to produce these droplets and then deliver to a specific locations in a on-demand manner. So wherever we want, wherever at what locations we want, we can actually basically deliver these materials along with cells or without cells or with biomolecules in a non-contact manner, in a or in an on-demand manner to build complex heterogeneous tissue structures. Now let us see what are the different milestones in this inkjet bioprinting, because this technology, it has started also in almost like 1980 80s. After that, people have started using inkjet printing technology. Before that, the inkjet printing was very much used for printing, like in the press or anywhere or in offices, mostly for printing like on piece of paper. But then this with the then few of the scientists, they found that this printing technology can be very well used for printing biomaterials, biomacromolecules, even biomolecules, and also even cells. So we'll see that. So it started in around 1988 when Klebe and his group, when they first found out that the HP thermal inkjet printer can be very well used for printing biomaterials like collagen, fibronectin. So that was the very first attempt of printing biomaterials or biomolecules with the help of a inkjet 3D bioprinter or with the help of a inkjet bioprinter. So with the initial success of that thing, later and in 1997, Griffith et al., they have fabricated scaffolds, 3D scaffolds with inkjet printing technology using biomaterials like PLGA, PLLA. Both these are synthetic biomaterial. So they have used this biomaterial to print 3D structures with the help of a inkjet bioprinter. Then followed by that in 2000, Macbeth et al., they use 3D by inkjet 3D printing technology for patterning bioactive proteins, and then they found that the proteins can be very well patterned on a 2D structure using this 3D printing using this inkjet printing technology. So with this success, later on in 2000, the same year, Goldman, they patterned biomolecules like denatured DNA with a HP inkjet printer. Right? So they patterned the DNA. So for DNA patterning, also the inkjet 3D printing technology was used in 2000. Then in 2003, this researcher called Wilson and Boland, they also used an inkjet printer to print a biomolecule called biotin with the help of this inkjet printing technology. Then the same, followed by that year, Boland and his colleagues, they first attempted to print a living organism through an inkjet printer. So they used modified HP printer, or basically they modified a HP inkjet printer to suit printing of living organisms. In this case, they use E. coli organism for printing. They found that after printing, this organism is very much in living conditions. So this printing technology doesn't cause any damage to the organism, and that organism was in a live condition after printing. So that was the very first attempt of printing living organism through an inkjet printer. Followed by that, in the same group, they bioprinted mammalian cells with the success of that living organism like E. coli. They used now then they printed mammalian cells with the same printer in 2005. Then there are various attempts to bioprint cells, proteins, even different biomolecules like DNAs and other things. So in 2009, then Zhu and his colleagues, they bioprinted half-heart structure. So that was the very first attempt to print a tissue structure. Like in this case, it is a half-heart tissue structures with this technology. And then in 2013, the inkjet 3D printing technology was used for printing heterogeneous cells, where 3 different cell types were printed in a single construct. And then that was the first attempt where this research group, they have shown the possibility of printing heterogeneous 3D structures containing multiple types of cells with this inkjet 3D bioprinting technology. So this is the overall important milestones for this inkjet 3D bioprinting technology. But then after that, there are various research groups working on different applications of this 3D inkjet bioprinting technology. Now it is a very popular technology for printing cells, because in this case we can even also print single cells and we can pattern them. We can pattern different bioactive components like proteins, DNAs and other things. So that's why this technology is very much popular and it has been used widely for various applications. If you see the operations of this technology, it creates the model one layer at a time by placing a biopaper and putting the droplets on top of this biopaper, then putting another biopaper on top of this. Because here in this case we use the droplets, so with droplets we cannot create a 3D structure. So that's why a support layer called biopaper in this case is used to develop the 3D structure. So the one layer of biopaper is put on the printing stage. Then on top of that, the droplets are being placed on top of that. So the droplets are patterned, and then the first one layer of biopaper is placed, and then there the droplets are being patterned. So in this case we have shown a spherical pattern, but any type of pattern can be created. Either it can be square, spherical, or whatever the design is required. Accordingly, we can create the patterns. And then the next thing is this: another layer of biopaper is put on top of that, and then so this is the first layer is done. Then another layer of biopaper is placed, and then again the same process is repeated, where the droplets are again patterned on this second layer. Similarly, the third layer can be another layer of biopaper will be placed, and then same the droplets will be again printed. So this is how a height of or a required height is achieved. And then what happens, this biopaper is a biodegradable substance, so it will dissolve or it will degrade by its own. And before dissolving the cells, whatever the cells those are there in these droplets, they will come contact with each other, they will assemble with each other, and then they produce the extracellular matrix. They will make up the tissue, right? So the biopaper will be dissolved here, but the cells plotted here, or the cells that is printed here in a particular design or particular pattern, they will fuse with each other, and then they will create the tissue. So with inkjet 3D bioprinting, this is how a 3-dimensional tissue structure can be fabricated. So this inkjet 3D printing technology can be described, or as we have seen that, say here, the liquid droplets can be generated, and those droplets can be ejected by a controlled manner, right? Now, this ejection of droplets or the generation of the droplets, that can be done by various ways, but there are two most popular techniques available for this droplet generations are either it can be drop on demand, or it can be continuous jet. So we'll see one by one both this technology. Now in DoD printed, what happens? The droplets are ejected when required. So whenever a signal is given to the printed, only that time the droplets will be generated. That's why it is called drop on demand, right? Now what happens? This drop on demand, the printheads are equipped with multiple printheads. That means inside this printing device, there can be multiple print heads attached, and each print heads again can carry multiple nozzles. So there can be thousands of nozzles can be connected to a single printing device, right? So that's how it is built. Now what happens whenever a pressure pulse or a signal is given to the printed, only that time the droplets will be generated. If there is no signal is given, the droplets will be not generated. We'll see how the droplet is generated in this kind of drop on demand system. Again, this drop on demand system can be developed, it can be divided into two different types based upon the pressure developing system. That means whenever the signal is given, a pressure pulse is created, and then the droplets are generated. We'll see that those two are thermal based or piezoelectric based. In case of thermal, the first one, what happens? In this case, suppose this is a nozzle, a single nozzle that is lined up in between with the printhead, right? So this is just a single nozzle. Now you consider this single nozzle. In case of thermal, a heater is attached to each of these nozzles, right? Now this heater, and then the nozzle is filled with the liquid containing cells, either cells, biomolecules, biomaterials. Okay, so those things are filled. Those things filled up the nozzles here. Now, whenever this heater is heated up, what happens? A bubble will be produced, because the heater is heating up the liquid material that is present within the head, within the nozzle, right? So the bubble will be produced. So the bubble will be produced, that will increase the volume, and it will then burst. So due to this bubble production, there will be a pressure pulse that is being created within this the liquid column, and due to this pressure pulse, the droplet will be produced. Because this bubble, what is the bubble? When the bubble is growing, the increasing volume, it creates a pressure on this liquid chamber, and then a droplet will be produced from this thing. Now depending on the frequency of this bubble production, the droplets will be produced one after another. In case of to prevent the flow of this liquid backside, there is a neck kind of geometry is created toward the inlet side. So that's why the liquid cannot go back towards the inlet, but it is being generally pushed through the outlet or to the orifice where the droplets are being created, right? In certain inkjet 3D printer, that thermal inkjet 3D printer, the heater can shoot up to 200 to 250 degrees centigrade. But what happens? This heating is so transient, because it is for so very small duration. So that's why the temperature of this liquid, that never reaches to that thing. The local temperature of the liquid can reach, or can be raised 4 to 10 degrees centigrade, not beyond that. So that's why this printing technology can be well for printing cells also. And there are studies where it was found that The cell viability is almost more than 90% cells are viable, or more than 95% cells are viable using this printing technology. So this was very well used for printing cells. And the for thermal DOD printer, the orifice or the nozzle diameter is somewhere around 50 micron. So the droplets of any size, anywhere between 30 to 80 micron size of droplets can be very well produced with this printing technology. But because of this very tiny, very fine orifice size like 50 micron, there can be sometimes the nozzle clogging. So we need to choose the process parameters accordingly so that the printing process can be smooth and accurate or efficient, right? So that's the principle of thermal DOD printing. Based upon the heating, the bubble production, the droplets are generated, and that those are being used. So this printing thermal DOD is very widely used for this thing for different applications. The next printing technology is piezoelectric crystals, piezo based inkjet printing, where what happens, again the same liquid column, inside the liquid column that a piezo crystals is attached. As piezo, as you know, this is a transducer. So whenever an electrical signal is given to the piezo, it converts into mechanical energy, so it starts vibrating. Due to the vibration, it creates a pressure on the liquid column, and due to this, the droplet will be ejected from this piezo, from the nozzle size, right? So here the based upon the vibration, the droplets are created, right? So here what happens, so here also the nozzle can be, nozzle diameter can be around anywhere between 18 to 30 micron, and so the droplets generated in this case can be lower than the thermal DOD printer, right? And here also this can also be very well used for printing cells. So as you can see here, the cells can be very well suspended within the media, in the liquid or the media or buffer, and then that can be, the droplets can be generated from this, where these droplets can also can also consist, the cells can also be there within this generated droplets. But depending on the frequency, if suppose the vibration frequency reaches somewhere like 15 to 25 kilohertz, that can be detrimental to the cells. There are few studies, they have found that there can be a cell damage due to if the vibration frequency in that range. So but if you can control or optimize the process parameters, like if you can select the vibration frequency to a certain range, then that can be very well used for printing cells. So this technology is also very popular for printing cells. Now printing different biomolecules. So both this thermal and piezoelectric crystals, both this printing 3D printing technology are used for printing biomaterials, biomolecules, cells, for either for patterning or for creating 3D structures or for different other applications. We will see some of the applications in the later slides. A few characteristics of this inkjet printing technology, if you want to understand, the first is because in this case the main mechanism of printing is the droplet generation. So microdropletization is an important characteristics of this inkjet bioprinting technique, where this inkjet printing technique ejects tiny droplets as the basic formation units. So it generates these droplets, and with those droplets basically we are creating the whole 3D structure. So this is an important characteristics of inkjet bioprinting. Next thing is the high throughput. Because of this high ejection frequency and high parallel throughput, what happens, as I already discussed, within the printers there are multiple nozzles, those are lined up. So parallelly we can generate multiple droplets, right? So in certain cases there are hundreds of droplets can be generated at a time, and the frequency of ejection is very high. So that's why it's a very efficient and high throughput method for generation of for inkjet for printing 3D for printing various structures. Another important characteristic is the non-contact nature, because the print head and the stage both are separated from each other. Okay, and they are not coming in contact anytime, and also they are not connected to each other by some materials also, because this droplets are these are created, separate droplets are created, discrete droplets, and they fly from the nozzle to the stage, right, to the substrate, right? So that's why the contact nature, that's a very good advantage, has very good advantage for various applications. So that's why this technology is very popular for certain applications. The other thing is the drop on demand. As we have seen that whenever a droplet is required, that time only the drops are basically generated. So the drop on demand technology is very important characteristics of this bioprinting technology. Thank you. The remaining thing we'll see in the next session. Thank you very much.